Afternoon, folks. Welcome to the Royal Society of Biology presentation this afternoon as part of Biology Week. We're really looking forward to hosting this event for you this afternoon. So whether you've had a long day at school, like me, and you're just finished for the day, or you've joined a STEM club to join our event, or wherever you are in the UK or around the world, a big welcome from the Royal Society of Biology. And it's been a real joy to put this talk together for you this afternoon. So I'd like to introduce our three colleagues this afternoon. We've got Helen, and we have got Parvez, and we've got Samantha, and they are all waiting online to give their presentations. After their presentations, there'll be a quick little interlude from me, and then I'll pass on to the next presentation. And in the last 10-15 minutes of the session this afternoon, we will have Q&A. So could I welcome our first speaker this afternoon, and it is Helen. Helen, are you there?
Yes, hello.
Hello, Helen.
Excellent.
And you're a third-year PhD student at the University of York.
Yes, indeed.
So you're near the end?
I only just started my third year, so I'm just slightly halfway.
Well done, you. It took me 4 years for mine, so but well done. It's a real test of resilience, the PhD, but a fantastic qualification. You've been researching social behaviour in male African savanna elephants.
Indeed, yes.
So I'm trying to find a link into each of our presentations. So here's my link for you, Helen. I've got some elephant poo.
Fantastic.
It's used to make paper, allegedly. Is that right?
Yes, yeah, sometimes. Yeah.
So that's my link for you this afternoon. But I know you're not talking about poo. You're talking about the change in their behaviour as we get older in elephants.
Indeed, yes.
Okay.
However, you say that, but if I actually start showing you my screen, you can see actually I am going to talk about poo a little bit, but not too much.
Over to you, Helen.
Excellent. Thank you very much. So I started university back in 2014, and I went there expecting that one day I was going to be working probably in a medical lab, maybe for a conservation consultancy. I certainly never thought that I would actually end up saying the phrase during my job, "This dung must be nearly an hour old. It won't peel properly." This is as weird as it sounds. I was trying to sample the DNA from the outside of a bowl of elephant poo. And when it's really fresh, that outer coating just peels off really nice and easily. But as the sun dries it out, it doesn't peel as well. So it becomes a lot more awkward to sample. So what is my job? It's not moving on. There we go. I am what's called a behavioral ecologist, which doesn't necessarily sound particularly exciting, but I'll break down for you what that means. So ecology is the study of relationships between organisms. Now, this could be food chains, looking at predator-prey relationships. It could be parasitic relationships where one organism actually lives off of another, or it could be relationships of others within your species, such as group behavior or parental care. Now, as a behavioral ecologist, I study the behavior of interacting organisms. So you can simplify this and just say that I am an animal behavior scientist. And this is my wonderful study species, the African savanna elephant. So specifically, as Andy mentioned, I am looking at the males and looking at their social relationships with one another. So this is the basis of my PhD. So for those of you who don't know, a PhD is a high-level university qualification. And to get here, I've had to do an undergraduate degree and then a master's degree. And after this, I will be fully qualified as an animal behavior researcher. So I'm doing this work at the University of York, and I'm collaborating with a charity called Elephants for Africa, who work in Botswana to try and find out more about the elephants who are living in the Makgadikgadi pans, and working with local communities to try and improve coexistence between the elephants and the people. Now, I'm not going to talk any more about the dung research because I actually had to cancel that. But instead today, I'm going to talk to you about the other work that I was doing while I was out in Botswana last year. So to start with, I'd like to introduce you to someone. So this is Siska, and he's a teenage male elephant, and he's about the age where it's time for him to leave his family group and go and join the world of adult elephants. So up until now, he's lived with his mum, his auntie, his grandma, his cousins, and they've cared for him, they've taught him how to find food and water, and when danger has presented itself, they've protected him. Now, in a family group, you'll find that the adult females form a protective circle around their babies, and just to keep them safe. And as he is aged, he's moved from being in the middle of that group to now being one of the defenders of the younger babies. But he still always had his mum and the others at his side. So my question to you today is, once Siska leaves his family, what should he now do if he's in danger? Now, luckily for Siska, just because he's left his family doesn't actually mean he's going to be alone. So male elephants will also form groups, but they're much more variable than the female herds, and which males are grouping together is constantly changing day by day. So say he's hanging out with these other 4 boys and a lion comes along. What should Siska do? Well, if you think about what's the first thing you do, if you're at school and the fire alarm goes off, your automatic reaction is that you look around and you see what's everyone else doing, and you probably do the same as what they're doing. So maybe that's what Siska should do. Look around and see what his friends are doing, and then copy one of them. But if so, who should he copy? So Johannes and Harry, only slightly older than he is. So they're more likely to be his friends, but they might actually not know much more than he does. They're just as inexperienced as him. Squonk is really grumpy. He's just a grumpy elephant. And so therefore he might be a better option for protecting Siska, but he might also charge at the lion. So if Siska copies him, he might actually be moving towards the threat, in which case it could put him in more danger. Colin is the oldest, and therefore he's the most experienced of the group, but he's also the biggest and therefore potentially the least threatened by a lion. So again, he might just ignore it, which if Siska then does that, it could again put Siska in more danger. So it would be just doing something inappropriate for Siska's own life stage. Another option is that he could look at what everyone's doing and then make his own decision. He may not copy anyone, or even all of his new friends may come together the way the females do and protect Siska as the youngest individual of the group. So understanding how elephants make these kind of decisions is actually really important to their conservation. We need to know how well they can protect themselves when they're in danger, but we also need to know if they're more likely to act defensively or attack a threat. And that's in case they come into contact with humans. So human-elephant coexistence is really, really difficult because elephants can be extremely dangerous when they want to be. So if we know how male elephants are likely to react to danger when they're in a group, we can protect not just them, but also the people who live around them. So how do we actually go about finding this out? So I ran a set of experiments in Botswana called playbacks, where we basically play sounds to a group of elephants and we see, do the young ones copy the older ones, and do the older ones protect the young ones? So this was our plan set up, what you can see here. So we had the speaker was on the ground here, we had the car was a short distance away so that we could sit in the car and watch them from a safe distance and film them. And elephants all nicely in a little group. However, these are wild elephants and we were based in a national park, not a laboratory. So often the setup actually looked a little bit like this. So we might not have a clear view of the elephants, the distances might be quite variable. And actually by the time we reached wet season towards the end of my fieldwork, it actually looked at something a little bit like this. Now, when we think of scientific experiments, we often tend to think of labs with really controlled conditions where we can measure everything, we can make every repeat of the experiment exactly the same. This is really, really rare in ecology. Because there's just so many things that could possibly change, because we're looking at species relationships in their natural ecosystem, we can't possibly begin to control everything that's going on. Things like the weather, things like whether the elephants were actually attacked by a lion that morning, and therefore they're already on high alert for lions. All we can actually do in ecology is basically make every experiment as similar as possible, and then we have to analyze our results really, really carefully, often with some very complicated stats in order to be very confident in our answers. So these were the sounds that we were playing. So we had a local species of dove, we had a lion, and we had a human. Now the dove, of course, is not actually threatening to an elephant. This was purely being used as what we call a control stimulus to ensure that any reactions that we see to the lion and the human sounds are not just responses to the speaker or the presence of the car, they are actually fear responses relative to those noises. So, and I hope this works, we're going to try and see what this actually looks like. So here I've got a clip from a lion playback, and I want to show you the kind of data that I'm interested in. So I'm looking to see whether elephants are more likely to look towards each other and move towards each other after we play the sound than they were before. So what you're going to see down the bottom here is notes for every elephant showing what each one is doing relative to every other elephant at each point in time. So if we take bull number 5, so it's this guy here in the middle, You can see at the moment he is looking directly at these three on the left. He's side on to this guy right in the back that you can probably at the moment just see his tusk. And he is looking directly away from this one on the right. Now, as it plays, the labels are going to change. I suggest maybe just pick one elephant and watch him so you can see how they change rather than trying to watch them all at once. So here we go. Okay, so hopefully that should have given you a reasonable idea of what it is that I'm actually looking at. So I've got 48 videos like this in total, so I've got 16 per type of sound. And from this, we can sort of see who moved first, who looked at who, and potentially who copied who. So for example, here, it's fairly clear that bull 2 did not move in response to the speaker because he was already leaving as the stimulus started. But bull 1 left about 5 seconds after it started. So we now need to look at all of our other data and try and work out whether it's more likely that bull 1 was moving in response to the speaker, or if he was more likely to be moving in response to bull 2's just following bull 2. But before we look at any data, I just want to make sure that we're all familiar with the type of graph that I'm using. So this is called a box plot, and it's a way of comparing the distribution of data between groups. So it breaks our data down into 5 values. So we've got our highest value up at the top and our lowest value at the bottom. We've got our median value. So this is, we have half of our data are above this point and half of our data are below this point. And then we have our quartiles. So we've got a quarter of our data are below the lower quartile and a quarter of the data up above the upper quartile. We might also have a few outlier points. So these are plotted separately because they're just data values that don't seem to fit with the rest of the distribution. Now, if all of our groups are the same, we'll see something that looks like this. So we've got a lot of overlap between our boxes. If they're really different, we'll see something that looks like this. So you can see that this group here produces values that are far smaller than this group up here. And then if we get something that looks like this is where things get interesting, because here we've got only small differences. So this is where we have to do our statistics in order to know whether these are genuine differences between the groups, or if actually they might just be to do with just random chance, just small amounts of variation that don't actually mean anything. So I haven't yet had a chance on these data to do the actual statistics. So everything I'm going to be showing you is just based on what we can see from the box plots. But I think hopefully we should see something interesting. So here we go. So this is looking at the looking directions, and this is before we've played any sound. So at this point, they haven't heard any noises at all. So what we've got is 3 different looking directions. So we've got looking directly at another individual. We've got them looking side on to another individual, and we've got them looking directly away from another individual. And for each of those, we've got 3 different colors. So the purple is the control stimulus, so that's the dove, green is the lion, and yellow is the human. Now, what we can see from this is that the elephants are spending more time before the stimulus looking directly away from one another than they are looking directly at one another. Now, what this suggests to us is that actually what the elephants are doing is they are setting up in little groups that all stand together, but they face away from each other. So they're constantly scanning for danger and they're watching each other's backs. So they're helping to protect each other. But if we now look at what happens after we start playing the sound, then we see something that gets a little bit more interesting. So I've split the data up a little bit more again. So now, as well as comparing the types of experiment, we can also look at the ages of the elephants involved. So on the left here, we've got elephants that are looking at, side on, or away from individuals who are younger than them. In the middle, at, side on, or away from other elephants who are the same age as them. And over on the right, at, side on, or away from elephants who are older than them. So we still see that the elephants spend less time looking directly at one another than directly away from each other. but they are more likely to look at each other during the green lion experiments than they are the control or the human. So if we look at this particular set, so the ones where they're looking at a younger elephant, it seems like the older males, like Colin and Squonk from the start, are maybe looking out for the younger ones, like little Siska, and just checking that they're safe. And over here, to a slightly lesser extent, but definitely still there, the youngsters do seem to be looking towards the older ones, either to just see what to do and copy them, or potentially for protection. If we just look at side on and look away, we actually don't see a massive difference between the colours. So the control here is relatively similar to the lion and the human. So we're not necessarily seeing a huge difference between our types of experiment. On the other hand, if we look at the movement directions, we do see that big difference between the experiment types. So you can see almost immediately that we get no movement at all for a control stimulus. We can't see any of those purple bars, they're too small. The green ones, the lion ones, are a little bit bigger. So we've got some movement during a lion stimulus, but still not much. For the yellow ones, the human ones, much bigger boxes. So what we're seeing is that we get a lot more movement in a human stimulus than a lion, and a lot more in a lion than a control. We also see, if you look at the direction that they're actually moving, they're more likely to move away from each other than they are towards each other. But there doesn't seem to really be any difference between the age categories. So if we only consider the looking directions, it would seem that there's not a huge difference between the stimuli, but there is a difference between the ages. Whereas if we look at the movement directions, we see that there's a big difference between the stimuli, but no difference between the ages. So in order to spot the nuances of their behavior, we're actually having to look at everything together and compare multiple different behaviors at once. So overall, what have we learned so far? So we found out that before a stimulus, when they're not feeling threatened, the elephants are protecting each other and looking out for each other the whole time. After a lion stimulus, which is something that can harm a young elephant but is of limited danger to an older elephant, the older ones are looking out for the younger ones, and the younger ones are looking to the older ones potentially for protection. But when they're seriously threatened, they seem to just be panicking and running, usually away from each other as well as away from the stimulus. And they don't really necessarily seem to be looking at each other first. So all just running off in a bit of a panic and just not really checking to see if their friends are okay. So that is as far as I've got so far. And I'm sure at some point I will be able to tell you more once I've actually been able to do the stats. But for now, that is it. So thank you very much for listening. And I also just need to say a quick thank you to my field team for helping me with all the research, my supervisors at the University of York and at Elephants for Africa, and to NERC for funding this research. And of course, a massive thanks to the wonderful, wonderful elephants of the Machari Pans National Park for allowing me to run these experiments on them. And thank you guys for listening.
Thank you so much, Helen. That is fascinating insight into your work. And isn't it amazing how many people are involved behind the scenes as well in your thanking at the end? A PhD isn't just about one person, it's about a whole team of people all working together. So thank you very much. And also from a science teacher's point of view, looking at your data as well, thinking about how we as teachers help students and schools to develop a wide range of data management techniques is so important. But thank you very much. You're going to have some great questions later on, I'm sure you are. Thank you very much. Can I introduce now Parvez? Parvez, I've got a little introduction for you.
Sure.
You are fascinated by everything since forever.
Yes.
And you choose to spend your time researching natural organisms in terms of form and function. Well, one of my favorite organisms, Parvez, is my moss balls. Okay, I was given this by Maisie Smith in my year 12 class, and it's amazing, these moss balls. And I'd like to know all about their form and function later on in the Q&A. So over to you, Parvez.
All right, so I should just quickly say hi and thank you for this invitation. My name, as you know, is Parvez. I'm a reader in mechanical engineering at the University of Edinburgh in the UK, and today I'm going to speak to you about biomimetic design. I'll explain that in further detail soon, but biomimetic design is basically one of my main areas of research interest. But before I start speaking about biomimetic design, perhaps I could introduce you to what mechanical engineers do, which is, the meat and potatoes of what I do on a day-to-day basis. What we do fundamentally is we make things that move. So this could include, for example, planes or anything in aerospace, automotives, renewable energy technologies. If you look at the picture there, those blades will be moving, of course. A lot of work in robotics and in advanced manufacturing. If you think of 3D printers now, a lot of people are using those. Lots and lots of moving parts in 3D printers. Even your coffee maker has moving parts, and so of course we have to design things when we are ready to make these moving things. And of course, specialty materials is another area of interest for mechanical engineers, because of course when you load a material, it will move internally. And biology, this is the last thing that I'm saying here, is like that is a slightly unconventional thing to put down for mechanical engineers, but there are a few of us who have an interest in biology, and that is fundamentally because we are interested in the design aspects of biology. So you might be asking, well, we've got lots of really great cars, planes, trains, automobiles, etc. Why would a mechanical engineer, or any engineer in fact, need to understand or learn about biology? Well, there are quite simple answers to that. And one of those is that biology is extremely diverse. We do a lot of designing parametrically, non-parametrically, but we have boundaries. But biology seems to be almost boundless. There are all sorts of diversity that exists at every length scale. At the gargantuan length scale, for example, you will find there are ancient species of pterosaurs that had their crests at the front of their heads, and that allowed them to sort of turn in midair much more quickly than planes of today turn. A lot of the planes that we have, they'll have their fins at the back, but based on the pterosaurs, there are some researchers at the University of Florida, for example, who've been making aircraft with fins further at the front. There is a bit of a trade-off, of course, with stability, but it depends on what you're after at the end of the day. At the macro level, you might find bone is a good example to go with. There are lots and lots of different types of bone in nature. If you look at bone, it's a cellular structure. So it's like basically a compact material with lots and lots of holes in it. And what that allows you to have is a material that is stiff, it's strong, but it's also very lightweight. And this is generally a good thing. One nice example of bone-inspired engineering is the Eiffel Tower. So that's inspired by trabecular bone, from what I've understood. At the nanoscale, you'll have really interesting things. One example that I'm showing you here is a gecko foot. Geckos, as you know, they're these the lizards that sort of walk around on walls, on ceilings, upside down. And you do sometimes wonder how they're able to do that, because they don't have any wet adhesive or glue or anything like that under their feet. It's all dry. What's actually going on with the gecko is that it has lots and lots of hairs at the bottom of that foot. And then on top of those, it's got lots and lots of more hairs. And what that enables is an increase in the surface area of contact between the foot and the surface, which means secondary force interactions will be increased massively in situations like that. And I think a group at the University of Kiel, for example, have been designing these pads that they've been using. And I saw a picture of one of the Kiel University professors just hanging from the ceiling with one of these dry adhesion pads that he had developed over there. And then at the molecular level, you'll find that proteins and polysaccharides will have all sorts of shapes and conformations which have different properties. This image is, basically from spider silk. Spider silk is stronger than steel, actually, but it's definitely stronger than mild steel, I should say. And what it does is it arranges its molecules in a way so that it creates these aligned molecules that form nanocrystals that strengthen up the silk in general, and that actually increases the energy absorption capability of the silk. Now, we're talking about size differences here, and we've got a range of different organisms. Of course, diversity in biology is extant throughout a huge number of organisms. But of course, you can have diversity within similar organisms, like lizards, for example. You'll find if you take a feature of a lizard, like its claw, you actually find there's all sorts of different shapes, sizes, constructions, architectures that exist externally and internally when it comes to lizard claws. And there are some lizards that will climb trees. Those are called arboreal lizards. And there are some that don't climb trees. They burrow into the ground. Those are burrowing lizards. And if you look at the claws there in arboreal versus burrowers, you can see generally there's some kind of shape difference. So one of the things we did, for example, is we tried to find out if there is actually a shape effect on the ability for a lizard to climb more effectively or less effectively. And lo and behold, there actually is. We did some simulations. You can see penetration simulations, and we created our own equipment to be able to run interlocking tests for claws and that kind of stuff. And what we found is actually that the shape of tree climbing lizards, of their claws, is far, far superior to the shapes of the claws from burrowing lizards. So what we then did is we sort of applied this to bionic claws, and we made these steel claws. And we had one version that was mimicking precisely the claw shape of the tree climbing lizard. And then we used parametric design principles to vary the shapes of the claws, as you would as an engineer. And basically what we found was that the only claw that was really good for climbing, regardless of how many iterations you had with engineering, using engineering design principles, was the claw shape that belonged to the tree climbing lizard. So we have a very optimized structure there in the claws of tree climbing lizards, and it kind of is a testament to the fact that biology has a lot of answers and a lot of things going on that we need to understand better as engineers. And of course, ultimately, those are the claws that you're going to use for exosuits like Black Panther suits and so forth. Now, how biology can help engineers is not just, in terms of bionics, but of course, various other areas. One example I'm going to give you now is in robotics. We have been mimicking insects to develop flying insects, and there are different groups around the world who have been focused on developing insectoids, we call them. And one thing though is that the flight effectiveness is a function, or it's dependent on the size of the wing. And of course, if you have like an insectoid being deployed or landing somewhere, you don't really want massive wings, right? So there is a limitation in the way in which an insectoid can fly based on the size of its wings. But biology can help us there, of course, because if we could, for example, have larger wings that can unfold, for example, and then refold back in, then the deployment and landing is less of an issue. We can still use the same sort of space ranges that we're currently interested in. So one of the things that we've been researching recently is like wing folding. I don't know if you know much about origami, but origami is an ancient sort of Japanese paper folding art. And basically you can make all sorts of flowers and swans and birds and different things using paper, just as long as you can fold it in the right way. And what we find with, for example, beetle wings, and the one we're studying at the moment is this five-horned rhinoceros beetle, is that it has 11 folds in the wing that allows it to fold and sort of fold tightly and neatly under the carapace or under the shell of the beetle. So a system like that, if we can develop a system like that, then that's something that could be used for insectoids as a means of improving flight. How else? Well, I mean, if we stick to the subject of flight, well, we can, of course, consider how much energy needs to be put into flight. There's a lot of muscle power, you would imagine, going into insect flight. But actually, it may not be the case. I mean, if we have an insect-driven robot, insect-inspired robot, and it's flying away. It's using a battery, so it's using energy from a battery. But of course, as you're flapping, you're using a huge amount of energy. Is there any way we can preserve that energy so that that insect can fly longer, for example? And again, biology has these answers. These are just, if you look very carefully at those videos on the left, you'll see it'll pang there and then patang there. So those wings are basically bouncing back from a position of the maximum up and down strokes of a cicada in this case. And those guys are not even very good flyers. They're a bit clumsy. But anyway, the important point of note here is that the cicada wings that we've been studying, they store a lot of elastic mechanical energy, which means that if you are flapping, you don't actually need to use that much muscle power necessarily, because a lot of the energy that's coming back into that wing is from the first stroke. And that's kind of ingenious, and that's kind of something that we need to be able to apply to insect-inspired robots. Now, sticking on the subject of flapping wings, you can think about things like dragonflies. They actually flap between 6 and 30 times per second. The fastest flapper, I think, is the Scottish midge, which flaps more than 1,000 times per second. Now, aside from the fact that there's a lot of energy, et cetera, involved in that, you have other issues. It's like these wings are really thin. So one of the questions that I have is, how come they're not like tearing or fracturing, breaking or cracking during flight? If you're flat, you try and do it yourself. Try even six flaps per second with your arms. Very, very difficult. And you'll probably fatigue your elbows quite quickly, right? But what insects have in their wings is they have resilin, which is basically a rubbery kind of material. And this rubbery material stores a lot of the energy, and it dissipates energy very effectively. And as a result of that, these wings don't end up localizing strain energy, and they don't end up breaking during flight. Again, ingenious. So we applied that to bolted timber joints, and we actually found that if we just put a little bit of rubbery material, similar to resilin in properties, but put a bit of rubbery material, I think we put about 2.5 mil thick rubber around a bolt, we actually found we could increase the lifetime of a loaded joint by more than double. So there's, again, a lot of interesting stuff in biology that we can benefit from as engineers. So summarizing what biomimetic design is, well, the fun part is going out there, exploring, discovering new things. Once you've found something, a structure or an organism that moves in a specific way or has a specific interesting architecture that has a really amazing function, then you need to actually find out how it works. What is it that makes that tick, so to speak. As soon as you figure that out, you can start trying to mimic it or trying to find inspired ways of engineering structures. And once you've managed to do that, you have the potential to apply this to modern engineering technologies. Okay, so my last slide is to let you do some thinking. Some of the research we've done in the past has been focused on tree climbing fish. And these tree climbing fish actually also hop across the water. So they, in inverted commas, fly. So as I just sign off now, I'll just let you think about, what would you do? What would you copy as an engineer from a fish like this? And thank you very much for listening.
Parvez, hello. Thank you so much
No
for your presentation. That was really brilliant. Thank you. I don't know if you can see, but I've got Harry, my sulcata tortoise, here with me,
Yep.
And I was just fascinated by your pictures of your claws.
Nice.
Harry's got, he's only 6
Tortoises
months
are
old.
really interesting because they have hexagonal scutes in the center of their, and there's not that many structures in nature there's quite a few, but not that many structures in nature that are purely hexagonal, right?
It's
there you go.
he is he's 6 months old, and he's part of our
Nice.
reptile club at Highdown. And,
Nice. He's got a nice face as well.
he's got a friendly
He seems
face,
very
doesn't he?
peaceful. I like him.
Yeah.
You got a lot of stuff. It's great.
Biologists collect stuff. That's what we do. We're always asking questions about the world around us. Thank you so much. You're going to get lots of
No
questions
problem.
later on.
Sure.
Thank you. Can I now come over to Sam? Samantha, are you there?
Hi, yes, I am.
Hi. Now, my connection to you is I pulled my brain out.
That was great. I love that.
And I am fascinated by this thing. And I was shocked when I found it's the same mass as a bag of sugar.
Yeah, it's crazy, isn't it?
And
There's
no
so much to
wonder.
it.
No wonder all you students out there and some teachers do this at the end of the day. It gets a bit tiring carrying that around.
Okay, so hi everyone, my name is Sam and I've just finished my PhD in neuroscience from the University of Bristol. So I'm just going to be telling you a little bit about what neuroscience is and the sort of work that I do, and if you are interested in it, how you could potentially go about getting into a career in neuroscience. So slightly different to what the other speakers have spoken about, but hopefully still interesting to you all as well. Right, so what is biology? So I'm sure you're all aware, but just to recap briefly, biology is the study of all living organisms and their systems. So that can include things such as bacteria, fungi, plants, and animals. And of course, within animals, we are also humans, so we fall into that bracket as well. Neuroscience specifically is the study of the nervous system. So this can include the brain itself, as well as the spinal cord and all the nerves around the body. So anyone that researches or looks into any of these different aspects is a neuroscientist. However, myself and a lot of other researchers are particularly focused on the brain. So in terms of what I actually research, I look at a process called neurogenesis. So this is the formation of new neurons in the brain, and neurons are a specific cell type within the brain. In fact, they're one of the most popular cell types within the brain. So there are lots of different cell types, but this is the one that makes up most of the brain itself. As you can imagine, during pregnancy and during development, the entire brain itself needs to form. So there's a lot of neurogenesis going on at this point. However, neurogenesis can also occur in the adult brain, which is really exciting. In the adult brain, however, of course, the brain is already fully formed, so you don't need the whole brain to form itself again. And as such, adult neurogenesis is limited to very specific brain regions. One of these regions is the hippocampus, and this is shown here in the blue, and again in the blue box on the right-hand side. So I just want you to notice as well that within this hippocampus region, there's almost like a C shape and then a V tagged along at the end of that as well. So that's the hippocampus region that I'm looking at in terms of my neurogenesis process. In terms of looking at neurogenesis, you can actually see this happen within the brain itself. There are lots of different cell types within the hippocampus, and these can range from things such as stem cells. So these are cells that are unspecialized. They haven't yet committed to any particular cell fate, and they have the potential to form any type of cell within the brain. Obviously, these will exist as stem cells, and then based on any given cues around them, they can then form any specialized cell type. So in the case of neurogenesis, this will then form neurons, firstly immature neurons, and then over time will mature into fully mature neurons. However, like I say, this is based on different cues such as hormones and other proteins and other molecules. So this will dictate whether this stem cell forms a neuron or another type of brain cell. So why is this adult neurogenesis important? So as I mentioned, neurogenesis can occur within the hippocampus, particularly in adulthood, and this hippocampus brain region is important in learning and memory. So it's thought, it's not actually confirmed yet, but it's thought that this adult neurogenesis can help with learning and memory later in life. As you know, as you get older, you go through these different experiences, you make new memories, and it's thought that this formation of these new cells, these new neurons within your brain, helps you form these memories and then helps you learn how to reevaluate that situation should you ever face it again later in life. But what's really interesting is that although adult neurogenesis was discovered back in the 1960s, still sort of 60 years later, we don't exactly know what's going on with it at the moment. We don't know the exact function. We still don't really know how it works. So that really highlights just how little is known about the brain currently and how much more research there is to be done as well. In terms of neurogenesis, there's lots of different factors that can affect neurogenesis. These can include things such as aging. So this is just a natural process, a natural sort of part of life. So everyone will undergo it. As you age, obviously you do get older, your brain will start to deteriorate. So your level of neurogenesis will go down slightly. It's nothing to be concerned about. It does happen to everyone. You can also be affected, or your neurogenesis can be affected if you are subject to injury. So say if you've got a head or a brain injury, this might mean that your level of neurogenesis decreases, and this can be quite detrimental. Things such as lifestyle can also affect your rate of neurogenesis. So of course, there can be negative aspects that might lead to a decrease in neurogenesis, but there's also things you can do to make sure that you have an enhanced neurogenesis. So this is beneficial to you. So things such as having a good diet, maintaining a sort of healthy, a healthy lifestyle, so doing exercise, keeping fit, that sort of thing. So all of that can benefit your neurogenesis as well. And the thing that I'm most interested in is how stress impacts neurogenesis. So we all know that too much stress is bad for us, whether that's one big stress or whether it's a lot of little stresses over a long period of time, but stress can have a negative impact on neurogenesis. Interestingly, these stress hormones, so obviously once you're stressed, you get a hormone released, and this is that negative impact on neurogenesis. But these stress hormones also have a daily rhythm within the body. So these hormones are called glucocorticoids. As I mentioned, they're secreted following stress, but they also have a daily rhythm. So the hormone level is at its highest when we're in our active period. So for humans, that would be during the day, and for nocturnal animals, that would be during the night. And the level of hormone dips at its very lowest right in the middle of the night. Again, for humans, middle of the night, but for nocturnal animals, that will be the middle of the day. So this is a conserved rhythm of this hormone that is maintained in addition to the stress response as well. So this is something that I'm really interested in because I want to see how these stress hormones or these sort of daily rhythm hormones might affect neurogenesis. So in terms of the actual research I do, my group, the place I did my PhD in, we recently discovered that these hormones are required for neurogenesis to occur. So we found that obviously you need some of this hormone for the stem cells to differentiate into these neurons. However, if you didn't have any hormone at all, then you couldn't form any neurons. So that was really exciting. We also looked into the different levels of hormone. So whether it was sort of a hormone that replicates a stressful situation, so quite a high level of hormone, compared to whether it's a lower level of hormone that would follow that daily rhythm. So we found, interestingly, that even at these very low levels, so the levels that we would have in our bodies when we're asleep, even that would help promote neurogenesis. So it wouldn't be detrimental to it, but it would help for the formation of these neurons in the brain. And interestingly, we also found that to promote this neurogenesis, there is also a structure involved called cilia, which are very, very small hair-like structures. And these can help guide molecules such as hormones and other proteins, which, if you remember I said, are required to allow that stem cell to know that it needs to form a neuron. So it's likely that these glucocorticoid hormones, which have this stress response and this daily rhythm, are involved in making sure that these little hair-like structures, these cilia, have all the correct molecules, have all the correct hormones, and allow these stem cells to form these neurons. In terms of how I actually do my research, I can use these stem cells in the lab. So I often use these stem cells as a little ball of cells in a little flask, as you can see here. And I add the appropriate guidance cues and appropriate hormones and things to allow them to form these neurons. I can then look under a microscope to see these neurons myself. And this is an image of some of them here. So on the left-hand side, you can see a big blue blob with some little red lines coming out of it. So the blue blob is the ball of stem cells, and the red lines are the neurons all coming out of the stem cells. And on the right-hand side is just a zoomed in image of this. And again, you can see the lovely long red tails of these neurons. So that shows that you can successfully see these neurons forming from that stem cell itself. In addition to stem cells, you can actually look at the process of neurogenesis in an intact brain. So within the hippocampus, as I mentioned, this is where the adult neurogenesis takes place. You can look into this in a brain itself, and this is using a rat brain section. So if you remember that V shape I mentioned, that's what you can see on the screen in the two pictures. In that V shape, you've got the that sort of inner cell layer, so the red dots right in the middle of that V. That would be the stem cells. And in the top picture, you can see they're kind of red dots all the way lining that inside of the V. However, later on within that neurogenesis process, if you look in the bottom picture, the red dots have now formed red lines. So this is indicative of the stem cells turning into neurons over that neurogenesis process. So you can see this under a microscope, and it's actually really cool to be able to see neurogenesis happening right in front of you in real life. And just finally, I just wanted to finish on if you were interested in neuroscience and if you wanted to potentially become a neuroscientist or look into what you might need if you wanted to follow that career path. This is how I did it. So it's by no means how someone has to do it, but just it's my own experience. I did GCSEs and A-levels in biology and chemistry. I also did an A-level in psychology. So I think, I'm not sure what your syllabus is at the moment, but for me, I didn't actually cover much neuroscience in biology GCSE. That all came in biology A-level and then a little bit in psychology A-level as well. So I knew that I was interested in it, but I was interested in sort of wider science in general. So I then went on to study biochemistry at university, and I only specialised in neuroscience afterwards, sort of within my master's and then my PhD as well. So that's just one way to get into neuroscience if you're interested, but there's plenty of different ways. And if you are thinking about studying at university or anything like that, then have a look at the university requirements because it might be slightly different to what I've got here. Next, I just wanted to say that grades aren't everything. I know it's scary, especially being at school and everything is always based on the grades that you get, but honestly, grades are not everything. As long as you try your best, that really is all that counts. And if you show that you're interested in the things that you're looking at, and if you show that you've got that initiative, that's just as important. And again, I've mentioned as well, experience and initiative are really important. So where possible, attending things like this, so Biology Week Online, is such a good way to show that you're interested in biology, you're interested in whatever you are interested in. So if you can show that you've been doing these things, then that will really put you in a good place when you come to applying for jobs later in life or applying for university courses and things like that. And finally, just to finish, if possible, look for work experience opportunities around you. Like I say, grades aren't everything. And if you have that experience, if it's possible, then not only does it look good that you've got the experience, but it can also introduce you to what it's like to have a career in that field, whether it's neuroscience or something completely different. At least you'll have that exposure and you'll know whether that is something that you actually do want to go into. So just to finish off with that, hopefully that's been helpful to you. Yeah, thank you very much.
Thank you so much, Sam. Would you say we're all neuroscientists because we've all got a brain?
Definitely, I think so.
I think so. And long may neuroscience stay in the A-level curriculum. My A-level students love GABA. They absolutely adore GABA, and they're opening up chlorine channels. That's my A-level students. They love it. Thanks very much, Sam. Really appreciate your fascinating insight into your work, and well done on your PhD because you just got it, haven't you?
Yes, thank you.
Well done. If everybody could now turn on their cameras, that would be great, and open up their mics. That would be great. We've got 10 minutes now for some Q&A. There you are. You're back. Well done. Thanks to all of you, all three of you, so much for your time this afternoon and putting that energy and real passion that came through in all your research. And I think us biologists, we've got lots of passion. So big thank you. We've got some questions. I'm going to ask individual questions to each of you. Is that okay? Sam, I'm going to start off with you. Did you love science at school?
I did, actually. Yeah, science was my favourite subject at school. I definitely preferred biology more than the other sciences that I did, which I guess is the right answer here today as well. Yeah, so I always loved science at school, and I knew that that's something that I wanted to do as a career as well.
Brilliant, thank you. Parvez, did you always want to be a scientist?
No, I wanted to be a skateboarder. Still trying. But no, at school, I think I just enjoyed learning things, and progressing, learning new things, whether it was in Latin classes or whether it was in biology or chemistry or anything else. So yeah, I think science sort of just followed me, but actually I wanted to be a skateboarder.
Thank you very much. And have you developed skateboarding with your form and function?
This is a good question. I can tell you about materials for skateboards, but I don't know about that, okay, which is why I'm not a skateboarder.
Has it improved your riding of skateboards? Helen, what was the most interesting aspect? Sorry, what was the most unexpected discovery you made during your research in Botswana?
In Botswana, I think I would have to say, actually nothing to do with the elephants specifically. When I was recording those human stimuli, I said to people, imagine there is an elephant in your crop field. Can you please imitate what you would do to chase it away? And I sort of expected that actually most people, there would be a sort of fairly standard thing that most people would do, the same kind of thing. But actually I got really, really different things from different people. And that really surprised me. Like, so I had, you know, one person just whistled, one person was just like banging two pieces of metal together. One was literally singing a little song, which, like, if translated it from Setswana, it was basically just going, please go away now, elephants, elephants go away, it's time to go away now. One of them literally just grabbed a fence post and a piece of corrugated iron and started screaming and smashing them together. And that one just sounds like a machine gun going off, and it's terrifying. Like, but I sort of expected that they would all be quite similar, and they really weren't. And that really did surprise me.
How did the local people that you were working with, how did they see the research? Did they see that and value the research?
So I was actually mostly in a research camp, sort of fairly miles from anywhere. The nearest actual big town was my own, but that was still a 2-hour drive away. So far, my research has not really been discussed and disseminated with the local community. Once I've got the outputs, it will then be explained and distributed around the locals. But yeah, so far it was actually just the guys who I was working with in the camp, which I think they thought I was a little bit of a nutter for deliberately trying to scare elephants and being quite so obsessed with collecting bits of elephant poo. But in general, I think they were quite interested.
We're getting loads of questions coming through from everybody. Thanks so much, everybody, for your amazing questions. Sam, I've got one for you. Do you think your research has the potential for future treatment of conditions related to the death of brain cell, dementia-related, Alzheimer's?
Yeah, definitely. So a big thing, like I say, a big thing that I was looking at is the effects of stress as well. So there's so many stress-related disorders out there, such as anxiety, PTSD, things like that. And there's a lot of research out there, not stuff that I'm doing, but other researchers out there in the world that are looking into how these levels of stress, how they might affect making memories, like I mentioned. So this adult neurogenesis is involved in making memories later in life, and how you can manipulate recalling your memories in these traumatic events and how that might affect your neurogenesis and vice versa. So I do think there's definitely opportunities for therapeutics there.
Yeah, it really is an amazing piece of research that's so relevant to so many people and impacting on so many people's lives. Parvez, I've got a great one for you. For deep water exploration, have you done that in your time?
Not deep water, no.
Okay, well, you're
only to 25 meters, that's not deep
one.
enough.
Could the gills of fish be used as an example for gas exchange to have an easier transfer?
Yeah, I think so. I mean, there's actually quite a lot of biomimetic membrane-based technologies that have been covered by quite a few groups around the world. In over the last 10 years, I'd say. So there's definitely a lot of scope there. If you think about, like, the tree climbing fish that I put up, they actually like suck in a whole bunch of water into their mouths, and then they have that gas exchange through gills. They also are slightly amphibious, so they have it through their skin as well. So there's a lot of potential when it comes to biomimicry and gas exchange. But when it comes, I mean, to be honest, the big hurdle is manufacturing, because biological manufacturing processes are so advanced. Our manufacturing processes are not that advanced, so a lot of that small scale stuff is inspired engineering rather than mimetic engineering at this point in time. But perhaps with the growth of synthetic biology, that's going to change. We shall see.
Fantastic. Thank you. Helen, got one from you. Somebody's asked, why did you choose to study male elephants? And they've said, this isn't me, do you think female elephants would show similar behaviors?
That's a good question. So in terms of whether they show similar behaviors, I think the answer is actually no, because what we seem to be finding is that the males are a little bit, when it's something actually properly scary, they're a little bit sort of every man for himself, you just run away. But with the females, very much they protect each other the whole time. The reason I actually chose to study males is because we just know a lot less about males. If you watch things like David Attenborough, what you'll see is every time elephants are brought up, it's always female groups. And that's because actually females are just far easier to follow and study than males, because the males travel much greater distances. and it's a lot harder to keep an eye on them the whole time. So that's the reason we're looking at males overall. It's also in the Makgadikgadi Pans, where I'm actually based and where Elephants for Africa work. The population is only 2% female. It's a really, really male-dominated population. So actually, if you want to study one or the other, it's far, far easier to study males there, because you can actually fairly well guarantee finding them, which is quite rare to find the females.
Thank you very much. That was from Kendrick School in Reading, so thanks ever so much on that one. Very quick one from somebody from it's S panel. They've asked, Sam, is there a way of possibly freezing neurogenesis in a single condition like cryogenesis for revival later?
Yeah, I mean, so with stem cells, we have them and then we freeze them down if we don't need them, and then we can revive them again to make neurons later on. And that's something that I do quite often. So it's definitely possible. It's way more possible at the stem cell stage, so when they still have that potential to become other cells, whereas once they're already committed to either a neuron or whatever cell type it might be, then that freezing process, there's a lot more of that cell that could be disrupted in that freezing process. Yeah. So it's a lot easier to do it for stem cells rather than a fully developed neuron.
Thanks very much. And a thumbs up or a thumbs down from all of you on this one. Is experience more important than grades for your careers? So a thumbs up if you agree, thumbs down if you disagree. Experience, is it more important? Oh, Helen, what are you going for?
I would say I think experience probably is more important, actually.
Okay, why do you think that?
Mostly because if you're looking at large charismatic animals, it's extremely competitive. So what they actually look for is, are you willing to go out and find the opportunities to get experience with it? And also because fieldwork is actually just really, really hard work, have you had a go at fieldwork and decided that you do actually really enjoy it, and you're not just going to burn out within a couple of weeks and go, no, I hate this? Personally, I think fieldwork's the best part. It's great fun. But for some, it's really not for everyone. So I think experience and actually knowing that you like it is a major, major thing.
Cool. Thanks so much. And last question, what career can you do if you take biology? From Colton Hills Community School. So let's tweak that a little bit. What as a biologist, what would you like to do if you had any job? Sam, let's start with you. What would you do?
Oh, I mean, I just love neuroscience so much. I think I'd stay in that. But yeah, just being able to look at these brains, whether it's a human brain or human brain system or an animal's, and just understanding, I mean, there's so many different aspects. Obviously, Helen's been looking at behavior as well. Obviously, the brain is very much a part of that, dictating in the behaviors that groups of animals or single animals take part in. So there's just so many different aspects of the brain that we still don't understand. So just anything to do with the brain, really.
Parvez, what would you do?
Obviously neuroscience and elephants. Those are the, yeah.
And Helen, what would you do?
I would say that I'm very grateful that there's people like Sam out there who want to do neuroscience, because personally, I'm a very visual thinker, and if it's so small that I need a microscope to see it, I've struggled to be interested. Didn't necessarily need to go as large as elephants, but I personally think that I find behavior is so interesting, and social behavior specifically. I absolutely love it. I'd stay in social behavior.
Well, I think all three of you would make fantastic biology teachers in school. And I think you'd absolutely inspire a whole new generation. And we need lots more biology teachers. So if you're in a STEM club, thank your biology teachers. If you're in school, thank your biology teachers. We need more of them. So parents out there, if you want to come into biology, get in touch, because we need more biology teachers to inspire some amazing colleagues like we've had tonight with Sam, Parvez and Helen. Thank you, all of you, for your presentations tonight. It's been quite inspiring, and enjoy the rest of the day. Thanks very much. Cheers, everybody.
Thank you.
Thank you.
Bye.